Our next speaker, our third speaker, is Dr. TJ Higgins. Dr. Higgins is an Honorary Fellow at CSIRO Agriculture in Canberra, Australia. His major research focus is the application of gene technology for plant improvement. He's particularly interested in protecting food legumes from insect pests and enhancing the nutritional quality of plant products for feed and food uses. Most of his current work is aimed at increasing food security in Africa and India. Dr. Higgins was born in Ireland. His first degrees were in agricultural science from the National University of Ireland. He gained his PhD from the University of California, Davis, and traveled to Australia as a postdoctoral fellow at the Australian Natural University. Shortly after, he joined CSIRO Plant Industry, from which he recently retired as a chief research scientist and deputy chief. Dr. Higgins is a fellow of the Academy of Technological Sciences and Engineering and a fellow of the Australian Academy of Science. His presentation today is entitled BT Cowpeas Are Resistant to Podborer. Dr. Higgins, please.
Thank you very much. Bearing in mind what the time of the day is, and it's just shortly after lunch, I have put the take-home message on the first slide, so you don't have to stay awake for the whole thing. This is a typical area in West Africa where cowpeas are grown and where much of my work is focused at the moment. This is a rural area in Burkina Faso. Before I tell you a little bit more about the project, I should like to start off by thanking various people, particularly the people who are my colleagues working in West Africa. These are the cowpea breeders in Nigeria, Burkina Faso, and Ghana. Mohamed Ishaku, JB Tinegre, and IDK Aktople at CSIR in Ghana. I'd also like to acknowledge Larry Murdoch, who really was the person who got me involved in this whole thing. He's at Purdue University, and many of you would probably know of him. I'd also like to acknowledge my CSIRO colleagues, particularly the entomologists. And last but not least, and in red down the bottom there, I'd like to acknowledge the funders of the work, the Rockefeller Foundation, for starting us out on learning how to transform cowpeas, and more recently USAID for their funding of the gene transfer work. CSIRO has also contributed something to this as well. And I'd like to particularly acknowledge AATF and the Monsanto company for the BT gene and access to the BT gene. Monsanto have provided the gene that I'm going to talk about on a royalty-free basis for use of this gene in Africa. So, cowpea, an important crop. Many of you probably have never even heard of it, though. It feeds about 200 million people in West Africa, provides them with a good source of protein and a very good source of a good quality, low GI starch. Global production, somebody had it on their slide yesterday at 5 million tons. It's actually closer to 10 million tons of cowpeas are produced every year when you take into consideration places like Brazil and Myanmar and India, as well as Africa. It, of course, as everybody knows, it fixes nitrogen, reduces inputs for the following crops, and there is a high feed value of the stubble, although many people have made a very good case today and yesterday for keeping that stubble on the land. But it is currently used largely to feed the livestock of these people. But despite all these positive things, there are problems as well. Pests and diseases are major problems, and particularly pests are a huge problem in cowpea. They're very popular. This is a type of agriculture that's not so common in Canada or in Australia for that matter. It's a very different type of agriculture. The farmers are small holders, in this case often less than a hectare. The cowpea is often intercropped with crops like sorghum or maize. So it is a very different sort of agriculture. I said here some men. Mostly it's women who produce the cowpeas, but some men do too. And I've used this case here, the example of a man who is making a statement that was highly relevant to what I was interested in doing, and you can read that there yourself faster than I can say it. But the take-home message here is that although they can use insecticides, they would much prefer not to be using insecticides, just like people everywhere else in the world. So the cowpea podborer, this is a lepidopteran pest. It's a very serious pest. It's not the only insect pest of cowpeas, but it's probably the #1 pest. The moth lays her eggs on the young flower buds usually. The larvae begin their growth within the flowers in a nice environment, humid environment, but they develop fully on the pods and on the seeds. And yield losses in bad years can be greater than 50%, sometimes much higher than that. So this is a problem. So the options that are available to the farmers are not to use anything at all, just to use the natural resistance that's there. IITA and other breeders have spent a lot of time on selecting out germplasm that would give protection against the podborer, and out of 16,000 different accessions in IITA, none have been found to show any useful degree of resistance. So doing nothing is not really a great option. In that case, yield can be as low as 0.35 tons per hectare. It's gradually creeping up. Now some farmers get up to 0.5 of a ton per hectare, but that's still not really acceptable. If they use insecticides, they can greatly increase their yield, up to nearly 2 tons per hectare. So you can see the importance of insects there, just in that figure, in the difference between 0.35 and 1.7 tons. So this is where I came into this project about 10 or 12 years ago, with the possibility of using biotechnology to introduce inbuilt resistance into the cowpea germplasm. What would be the yield increase in that case? I was hoping for maybe a 50% increase in yield if we could use at least one gene against a major insect pest. Let me tell you about what has happened in the meantime. We decided to proceed down the track of introducing Bt genes, which had been used very successfully in corn and other crops, soybean more recently, cotton, of course, too. But there wasn't a cowpea transformation system available at that time. So it took us a few years to get that going, but eventually we have got it going. It utilizes dry seed to begin with, which we imbibe overnight, incubate with agrobacterium. We use agrobacterium to transfer the genes, and then go through a tissue culture procedure, most of which will probably not be of too much interest to you, but we have a protocol that works quite well now. It's not hugely efficient, but it's workable. We can get plantlets like those shown here with little roots on them in tissue culture, put them into a high humidity environment, and then transfer them into the glass house where they flower and produce seed in the normal sort of way. So there is a transformation procedure there which takes about 6 months and is about 1% efficient. 1% of the starting egg plants gives us transgenic plants. Low, but manageable. We use agrobacterium, as I said. This is a typical binary vector that we have used. It has a selectable marker gene in it here, neomycin phosphotransferase, and we have the Cry1AB gene shown over here on the other side with its own promoter, a promoter from Arabidopsis, the small subunit gene, and a 3' end also from the small subunit gene. So this is a typical binary vector that most people would use for plant transformation. So how we screen the plants, we have produced about 800 independent transgenic events so far with this Cry1AB gene, and field trials, as I'll tell you, about 30 of those 800 in various countries. But our procedure here is to look for high levels of expression, especially in the flowers, because as I said, that's where the eggs are laid, is on the flowers. This is the larvae up here, boring into it. We look for single copies of the transgenes. We make sure there are no vector backbone sequences present. We make the lines, the selected lines, homozygous, look for good phenotypes that are no different to the parent genotype. And then we do laboratory bioassays with Maruca larvae in the lab. And then finally, if all of these things are working well, we take those plants to field trials and start biosafety studies. So we've been doing field trials we started, our first field trials were in Puerto Rico under the US system of regulation. There were no regulations at that time in Nigeria where we wanted to do trials, no biosafety regulations. But in 2009, the regulations came into play, and we were able to do confined field trials, first in Nigeria, in northern Nigeria, and then later in Burkina Faso, southwestern Burkina Faso, and then in northern Ghana. And this year we have started some trials in Malawi as well. So we've been doing these trials now since 2009, testing the various lines that are the best lines that we could produce in the lab. As I said, regulatory guidelines are very important. They were first introduced in West Africa, I think, in Burkina Faso, but Nigeria, which is where we wanted to do the trials, because Nigeria produces a lot of cowpea, had allowed us to do field trials there in 2009, and we've been doing trials since then with different lines. One very important thing that is often forgotten, and is certainly forgotten by molecular biologists like myself, is that we often think that if we can produce a plant that is insect resistant, for instance, that the breeders might use it. But they won't use it, in fact, if there is a yield drag associated with the transgene, with the gene that we put in there. Despite the fact that it might give very good protection, if there's any yield drag, the breeders won't use it. So this is an early experiment that we did with a couple of promising lines to show, this is the parent line here that we transformed. It's a line from IATA. And here are two promising lines that were doing very well in the confined field trials in the different countries. And you can see that there is no yield drag associated with the transgene. This was a very important point for us with the plant breeders. This is an example of one of the confined field trials in Nigeria in 2012, showing over here on the left-hand side a transgenic line and the non-transgenic parent line over there on the right-hand side. I hope you can see that there is lots of pods on these plants here and hardly any pods at all on that plant over there. So this is an example of a line that is showing promise. And here are two lines now showing you seed yield on the y-axis here in grams per plant. And along this axis, I'm showing you three lines, the non-transgenic parent line and two of the best performing lines in the field. So you can see that there is quite a big yield difference between the non-transgenic parent line and these two selected lines, 4- or 5-fold difference. More than I had anticipated, more than I had hoped for, much more of an increase than I had hoped for. That was in Nigeria just in 1 year in 2012. In Burkina Faso, on that year, the insect infestation was enormously high there in that year. So the non-transgenic parent line didn't yield any seed at all, basically, whereas the two selected lines yielded very well. There was no reduction in yield at all. And this is what that looked like in terms of seed per plant. This is the seed from the non-transgenic parent, from one non-transgenic parent plant, and the seed from 1709 and the seed from 1252D plant. You can see very dramatically the difference between them. So they're fully protected from the podborer. This is Mohamed Ishaku, one of the cowpea breeders in Nigeria, had already started backcrossing from one of these lines fairly early on. He could see that it was the kind of material that he wanted to introduce into his breeding program. And that's the same thing has happened in the other two countries, in Burkina and in Ghana. So now they have been introgressing the gene from that IT86 based line that I've shown you into their best germplasm, because they've been breeding and have produced better lines since we've been started doing this work, and they have now started to introgress the gene into their best germplasm. And this is one example here of transgenic and non-transgenic varieties now, rather than lines. This is a transgenic variety here, showing you the kind of yield compared to the non-transgenic sibling over here, which has most of its pods. There are no pods on this plant, so it's compensating and still green because it hasn't produced very much in the way of pods so far, and won't either. So these are now at the varietal stage. These are backcross 5, backcross 6 lines, and they've been tested over the last couple of years at multi-location trials in all 3 countries. This is one varietal trial, multi-location varietal trial in Nigeria last year, again showing you the transgenic and the non-transgenic with pods per plant, 21 versus 10, or seed weight in grams per plant, 22 versus 9. That's in Nigeria. So there's a twofold difference there. In Burkina, in 2015, again, higher infestation, so that the number of pods per plant on the non-transgenic are only 3 or 3.5 versus 21. And again, that's reflected in the seeds, seed yield, 31 versus 5 grams. So 4 to 5-fold increases in yield. So that's where we're up to with this. There are now further multi-location trials going on at the moment in all 3 countries, as well as new trials in Malawi, as I said. But we still have a few challenges. But just let me summarize by saying that we have now selected one line that has very good protection against the podborer, basically 100% protection, which yields as well as the parent line in the absence of insects. As I said, a very important point for the breeders. We're now testing these varieties still at multiple locations in all 3 countries this year as well. That'll be the 3rd year of multi-location trials. There are still things to do, of course, or other things that are going on. Biosafety studies on these lines, of course, are also required for the regulatory authorities in each of the countries. There are colleagues who are working on the consequences of outcrossing. Cowpea has one related species in Africa, and it does outcross to it. So there is a possibility there that we need to know what the consequences of that outcrossing would be to satisfy questions from the regulators. The consequences for non-target insects, that work is also going on. I'm not directly involved in these work. These are being done by my colleagues in Africa. And of course, the big one is community acceptance. That is still an unknown. We don't know whether we will be able to release these to the farmers. All the surveys that have been done by the social scientists would say that the farmers are keen to have this, and since the farmers are largely the consumers, I'm still hopeful that this will be accepted and that we will be able to get this material to the people that need it. The work that I'm currently doing in Australia is to add a second gene to reduce the risks of pod borers developing any resistance. So this is an important part of insect resistance management. By having a second mode of action, a gene with a different mode of action, will greatly reduce that risk. So that's technically what I'm doing at the moment. And we even have a logo for this line, almost ready to release. Thank you very much for your attention.